Now I'm gonna switch my my topics to kind of a basic scientist. I'm not MD clinician, but I'm a basic scientist. You know, but there probably as a physician, you guys probably realize that the disease is kind of really complicated. It's not just the single factors causing disease, but there are lots of genetic factors, environmental factors. But it comes down to actually basic scientists. We need to kind of dissect what's happening, what's going on in cellular and genetic levels. That's what I'm doing. Okay, I'm interested in one of the cellular specialized cellular population is called interstitial Cajal. And my groups in Reno, there's quite a big smooth muscle, gastrointestinal smooth muscle group. We working whole department, more than like around 100 PhD MDs. We working for gastrointestinal motilities. So as you know that the intestinal motility is regulated by these entering neurons. But these entering neurons interact with the two different interstitial cells. One is called ICC, and the other one is called PDGF alpha cell. And these cells are eventually regulating the smooth muscle cells, which are the final effectors make muscle contract and relaxation. So these three cells are ICC and PDGF alpha, and the smooth muscle cells, they are physically they are linked together. So we call cell, this is called by initial, the SIP, syncytium. So they create unique electric environment, this environment regulating gastrointestinal motilities. Okay, I'm interested in this talk. I'm going to focusing on this ICC. And there is a different population. They are located in different locations. Basically, these ICC cells are complicated than we previously thought. It's not just single population. Okay, so the outer layer GI tract, there is a serosal layers, epithelial serosal layers, but right under subserosal layers there's an ICC cell. It's called ICC-SS, subserosal, and also the longitudinal muscle layers. There's a intermuscular ICCs located here, and then between the circular and longitudinal muscle layers, also there's a myenteric regions, which is kind of neurons run. This is called myenteric neurons, ICC-MY. And also circular layers, there's a ICC, intermuscular circular ICCs there. And there are a large animal like a human, monkey, and dog. There's a septal regions right above, between the muscle bundles. It's called ICC sept. And also there's a submucosal regions here. The submucosal regions here, there is a ICC. It's called ICC deep muscular plugs. So these different populations, they play a different role, and these ICC they are major component actually regulating gastrointestinal motility. Now let's learn a little bit more about ICCs. So ICC are pacemaker cells. They are generating slow wave activities which are regulating GI motility. And their ICC cells are expressing the tyrosine receptor. It's called KIT. This KIT tyrosine receptor exclusively. And these kits are required for main development and maintenance of these old ICCs. And then if you ever lose or damage your ICC, they are actually directly linked to gastrointestinal GI motility disorders. Such as gastroparesis or intestinal obstruction. And ICC has been studied for last decade and generating over 2,000 publications. And this lead to discover unique ICC markers. One of them is a KIT, and the other one is recently found in calcium activated chloride channel. It's called TMEM16A or ANO1. But however, all the genetic informations and gene expression and also underlying molecular mechanisms regulating ICC depletion GI has not been discovered. So if we understand all these mechanisms and this, if we can identify all this genetic information, then probably we can develop some therapeutic drugs which we can reverse some gastrointestinal disorders. Okay, so we studied, but one of the problems with the studying ICC was, when the originally ICC was found, the people thought this is a neuronal cell. But now it's different from neurons, although they're doing kind of a similar job as a neuron. So ICC is a specialized cell, but it's really hard to study because we cannot identify the cell in vivo. So we developed kind of a GFP-labeled ICC cell mouse line. Okay. So we hook up with this GFP, like green fluorescent protein, and then we insert into KIT locus, and then we generate KIT COP GFP. We call KIT COP GFP this mouse line. And then here, and then we looked at the ICC cell in this fundus. The fundus, two different ICCs is running, muscular, circular and long-term muscle layers, two fundus, kind of long spindle-shaped ICCs we found. They all nicely labeled. And then we co-label with the KIT antibodies, and then they are co-localized beautifully. So, and also we looked at the all intestinal regions and two different populations. This is a muscular ICC, and this is a myenteric ICC, and this is a merged image here. But they all labeled. So this mouse was a great marker for study GI ICC some defect. And it's been known when the smooth muscles develop hypertrophy, the ICC is disappeared. And we studied intestinal obstruction for several years. What is the intestinal obstruction? Intestinal obstruction, a condition of significant mechanical impairment or complete arrest or passage content through the intestine. There are simple and strangulated intestinal obstruction, and also it can affect large and small intestine both. And there are acute and chronic obstructions, and also mechanical and then pseudo obstructions. And there are most intestinal obstructions caused by abdominal adhesion or tumors, interception or stitches and neuropathy, some neuronal damages. And the human studies show that the loss of defect ICC associated with the intestinal obstruction. So we made a mouse model again, because mouse model is great because they mimic mouse, but we can control, which now we can do human studies. So we surgically insert plastic ring in the at the end of ileum. And then this mouse is develop hypertrophy. So this is a mouse, and then we put the plastic ring. After 2 weeks, the muscles are hypertrophied. And then this is a kind of cross-section images showing here. So there's two muscle layers, the circular muscle layers and long-term muscle layers. And this is myenteric regions here. So this mouse, also we have a mouse model, has a labeling specifically smooth muscle cell with a cytoplasmic GFP. We're showing here all these smooth muscle. And also nuclear GFP, we can label the PDGF alpha cell only. So the nuclear label all these PDGF alpha cell. So these are double transgenic mouse labeling smooth muscle cell and then PDGF alpha cell. And then we can see the muscle cells are hypertrophied. These muscle cells are hypertrophied. And I didn't have time to show data, but we found that when the smooth muscle cells are hypertrophied, and these muscle cells are de-differentiated, and then they become PDGF alpha cell. So that's why smooth muscle cells are kind of very interesting. Somehow they can maintain stem cell kind of properties, and then they some pathological condition, they are go back to original precursor cell, and then they can divide. That causes a problem actually. And then after 2 weeks, once they develop hypertrophy, and then we can study what happened in recovery process. then we can remove the ring. And this is what we did there. We call a recovery surgery. This is a ring after 2 weeks, and hypertrophied here all the small intestine, and then we remove the ring, and then surgically closed. And then this mouse is showing that they recovery. The question here, ICC cells are, if they can regenerate or we know the ICC cells are degenerate in some pathological condition. But if the ICC can regenerate, and that's what we did there, we found yes, there's degeneration and regeneration. They are very dynamic cells. So this is ICC-MY, the myenteric ICC between the muscle layers. These ICC are generating pacemaker current. And this is what happens. So we label with the IC-specific, two different IC-specific markers. One is KIT and the other one is ANO1. So the control mouse has a nice networking ICC-MY. But after partial obstruction surgery, 15 days, ICC kind of disappear. They are degenerated. But when we remove the ring, they molecularly is coming back. This is after 1 week and 2 weeks and 4 weeks. After 4 weeks, the full ICC is coming back. And we looked at the different population the same way. This is IC-DMP, deep muscular plugs. So this is a control mouse. Nicely, they are integrated into the muscle layers. And then, but the partial obstruction, all these IC-DMPs disappear, just like a human pathological things we saw. And then after recovery surgery, day 1, and then 1 week and 2 weeks, 4 weeks, they are fully turned. So ICC cells, we confirmed the ICC cells they can degenerate in obstructional condition, but they can regenerate when the obstruction ring, the cluster ring was removed. And then another pathological example we're studying is diabetic gastroparesis. Gastroparesis, as you know, is a delayed gastric emptying. It's a disorder that slow and stop the movement of the food from stomach to intestine. And it's affecting people both type 1, type 2 diabetes. As Dr. Pasanka mentioned, that their obesity, about 1 out of 10 people has 70% has obesity in this country. So like 1 out of 5 has obesity. But out of that, the obesity people, they are the great kind of correlation with this diabetic gastroparesis. So about 1 out of 10. Total 29 million people in U.S. has a diagnosed or pro-diagnosis by this diabetes. Diabetes is a loss of glucose control, and the glucose level is increased in blood level, but the cells are dying, they're starving because the glucose cannot get into the cell because the control mechanism is destabilized. And also, there is a decreased ICC. Consistent findings suggest that the animal models and humans, they are related with the severe gastroparesis. Okay, and recent studies show that the epigenetic regulator is one of the class is a microRNAs. MicroRNA can regulate all basic cellular process, cell proliferation, cell differentiation, and apoptosis. So microRNA is kind of a great candidate for ICC depletion in this gastroparesis. So again, we use the mouse model. This is a leptin OBOB mouse. This mouse has a mutation in leptin gene, which is kind of sensing the fullness of food when you eat. And this mouse is eating excessively, this mutant mouse. So this mouse, this is the red is a control mouse and this is OBOB mouse. So at 4 weeks old mouse, already this mouse is larger than a control, and then they reach the top at around 12 weeks. When we measure glucose level also increased and is up to 350, is high, showing that hypoglycemia here in this mouse. And then we generate with this OBOB mouse model to this CARB-GFP, which can label ICCs with the GFP. And then we made this translating mouse, CARB-GFP, and lab OB mouse. And this is the mouse we generated. It's been published in 2010. And this mouse, we show that here, this is a non-diabetic control. This is all, this is the jejunum, and there is all ICCs, they are nicely organized. However, the diabetic mouse as a ICC was disappear. And the same as colon here. Colon has a different population, but the control, again, all ICC were there. But in diabetic mouse was ICC is kind of disappeared. And then we looked at the protein expression in KIT, and this is a control jejunum and colon, and this is diabetes. The KIT gene is disappeared. And then we looked at and then we isolate ICC, because one thing is nice things about mouse models. We can isolate a single cell population because all tissue data we can rely on all the pathological changes. So we isolate the ICC cell based on GFP expression using a cytometric method. So we isolate the single cell population ICC from control mouse, non-diabetic, and then diabetic mouse. And then we sequenced all the microRNA genes. And then we got about 2 million sequence, and then we analyzed all microRNA expression levels between 2. So we isolated from diabetes, this is colon, from colon ICC and jejunum ICCs. And this is a kind of heat map showing all the microRNA expression levels. So red is highly expressed, and then green is kind of not much expressed. So we found that one single microRNA is highly expressed, most highly expressed in ICC, which is kind of this is very interesting. So this microRNA is called microRNA 10B. So this microRNA appears to be they are ICC specific. However, these microRNAs, they are both in colon and jejunum ICC. They are all disappeared. So this mouse, there is this microRNA 10B is not only the microRNA required for this ICC, but the pathological marker we can identify this is some defect in diabetic ICCs. And then we analyze that what's the link from microRNA 10B to KIT gene we found in their disappear. KIT is required. If the KIT is not expressed in ICC cells, they are degenerated. Okay, so we analyzed that this 10B targeting protein using is called engineering pathways, IPA pathway. So IPA pathways integrated program. It can detect all the targeting protein based on all the previously published data and also bioinformatics, all proteomics data. They all integrate together, and then they can identify pathways. We found that several actually target genes interact with the target by microRNA 10B here. But the one thing is we kind of focusing in the KIT pathways. KIT. This is what happened in diabetes, by the way. This is diabetes. These are all proteins show that dysregulate in diabetes samples, human diabetes. Okay. And then we found that the two genes they are linked together. The one is the KIT, which is we want to something regulating by microRNA 10B. Okay. And then we found that this NCOR2, these proteins are actually negatively interact with the KIT. This NCOR2 is a epigenetic repressors. They are repressing gene expression. Okay. So, and then we looked at the NCOR2 protein in diabetes mouse. And then we found that these proteins, indeed, they are upregulated in diabetes, but the non-diabetes is less expressed, okay? Which is fit into our model, okay? So then we looked at the human samples, and we looked at first the KIT expression in this diabetic stomach, and sorry, this is a black is diabetes, and this is a non-diabetes. There's a reduction in KIT expression in diabetes stomach human patient. And also we looked at another gene, another IC cell marker, and ANO1, also kind of similar, there are reduction in diabetes. And then we looked at this is a messenger RNA expression, by the way. And then we looked at protein expression. These Western blot show that the protein also indeed they are decreased, they are reduced in diabetic patient. We also looked at the NCUR2, which is kind of a repressor for KIT. And we found that this NCUR2 is induced in diabetic patient. And we finally look at the microRNA expression in this human patient. There is a microRNA 10 base great reduction compared to other control microRNA show not much changes. So it's a great fit with our mouse model. And the next question we had is a microRNA can really control ICCs and then ICC depletion. And so we generated microRNA generate enzyme knockout mouse, but we don't want to knock out these microRNAs, all smooth muscle tissue. Okay, so we have a Cre mouse line, Cre recombinase mouse line specifically expressed in ICC because this mouse, this KIT Cre gene is under control KIT gene. Okay, so we generate transgenic double transgenic mouse line inducible KIT Cre mouse with a dicer lax mouse. And when we feed with the tamoxifen, they knock out specifically microRNA generating dicer enzyme in ICC. And this is what happened. So after 2 weeks, this mouse couldn't survive, they die. Because of this, all GI tracts is muscles degenerated, and all muscles is dilated. And then they couldn't survive, they die. And we look at the ICC in this knockout mouse, dice knockout mouse. This is wild type in GI genome, all the ICC cells there. But in knockout mouse, all ICC is gone. So clearly it demonstrates that the microRNAs, they are necessary for ICC development and maintenance in gastrointestinal tract. And then we generate microRNA. This is kind of global knockout. When we knock out all microRNA, ICC couldn't survive. The mouses die because all the ICC gone. And then we want to knock out only the dominant microRNA, ICC specific microRNA 10B. That's what we did there. So we used the same Cre mouse line, and then we cross with the microRNA 10B lax mouse. And we knock out microRNA 10B, only knock out 10B in ICCs. And then we didn't see great kind of a pathological defect, but however, when we look at the ICCs here knockout, we saw the same thing as we see the diabetes stomach. So ICC cells are mysteriously disappear, which show that this microRNA 10B is required for ICC maintenance and development. Okay, so this is our model. So this, so normal ICC microRNA 10B is highly expressed. This microRNA is knocked down targeting this NCUR2, the transcriptional repressor. However, in diabetic condition, this microRNA is shut down. And then what happened is that the NCUR2, it's been known, actually published NCUR2, they are interact with the histone deacetylase and the DNA methyltransferase. These are the players actually for making heterochromatin. They shut down the gene. These histone acetylase, histone deacetylase, they remove all the acetyl groups, and then they put the methylation on all these histones. So when the histones methylated, the gene is shut down. Okay, so this is what happened in diabetic ICCs. So just wrap up the summary. So I show that the ICC cells are very dynamic cells. They can regenerate degenerate in obstruction model and also diabetes mouse models. Okay, but they can also regenerate in recoveries. So they really dynamic cell. So ICC cells degenerate as they lose KIT. So the kit is required for ICC maintenance. Micronized 10B is gone in diabetic ICC and then target the epigenetic repressor NCOR2. So this abnormally increased NCOR2 may suppress the KIT expression in diabetic ICCs. And micronized 10B may restore KIT by NCOR2 in diabetic ICC. So what we're proposing here, so micronized 10B is not only a diagnostic marker, but it's a therapeutic target for degenerate ICC. And these are the people contribute to all these data, and this is my collaborators. Thank you.
Thank you, Seungil. Any questions? Okay, I just got one. So you showed a lot of changes in gene expression in ICC, in diabetic mice, and perhaps human. What if you control their diabetes well? If the hyperglycemia is not a problem. So, they start with diabetes and hyperglycemia, you control them, either weight loss or medicine or something, and bring the sugar to normal level. What's your guess? Do you think these cells will regenerate?
Yes, I think so, because as you proposed that we don't know what the GI disorders caused, but what we know that there's a genetic factors, and then also lots of as Dr. Solomon suggested, there might be some kind of nutrition factors can contribute. Yes, there is a epigenetic changes happening in our body. Actually, other scientists already demonstrate that. So what we're eating right now is really important, because this is going to be repopulate. First, what happens is that they're going to remodel your gene expression by some epigenetic mechanism. They change the histone modification and also methylation patterns. And this is going to be reshape gene expression. The change the gene expression eventually is going to repopulate cell population. This is going to be what happened in all the pathological process. So if the glucose level is back to normal level, it comes down to some epigenetic changes. They restore ICC later, because that's what we see that in obstruction models, when we put the ring, the ICC disappear. As soon as we remove the ring, they coming back,
right,
molecularly.
Thank you very much.